in here, right? Where there are exons that are protein coding, introns that are not. Those are all made part of the initial transcript, the pre-mRNA, and then splicing is removing those introns, the parts that don't code for protein. So we talked about initiation. How does RNA polymerase know where to bind to start transcription? Transcription factors, transcription factor binding sites help recruit RNA polymerase and get it going. Then there's elongation, which is boring. RNA polymerase does some sort of enzymatic thing that adds nucleotides onto a growing chain of RNA. That's all you need to know about elongation at this point. And then it has to know where to stop transcribing. And when it gets to the 3' end of the gene, there are some sequence motifs that tell RNA polymerase, hey, get the hell out of here. Stop transcribing. Fall off the DNA. So what we're talking about somewhere. It's in the processing and termination step. So there's one thing that happens during RNA processing, right? The primary transcript's made, start to end. It's got introns in it still, and 3 things happen. One thing I'm not talking about, except I'm going to say it right now, a 5' methyl cap is put on the 5' end of the transcript. The very start of the transcript gets a modification so that it doesn't degrade from the 5' end. The 3' end of the transcript gets a different modification, poly-A tail. So there's an enzyme at the very end of the transcript that just adds A. And as this slide says, maybe 150 to 200 A's in a row. So at the end of every messenger RNA gets poly-A, lots of A's. And while that's happening, while all of this processing is happening, the cap's getting added, the poly-A tail gets added, and splicing happens. So that's where we are today. Okay, at the molecular level, this is the essential structure of the intron. 5 different nucleotides. This is where consensus actually works. It's not just 75% of the time it's this letter and the other 25% of the time it's the other 3 nucleotides. G and U, 100% of the time, are always at the 5' end of the intron. So the first 2 nucleotides in the intron are always GU. There's some amount of distance, thousands of base pairs sometimes in introns. You get the branch point, a single A that's 100% conserved. It's always an A. And then some huge amount of distance more, well, in this case, as this figure says, 15 to 45 bases. The last 2 nucleotides are always AG. Right, so the splice sites that you might remember from classes way back when is GU, AG. Those are the flanks of the introns, always. GU at the start of the intron, AG at the other end. This is presumably how the cell knows where to splice. There's something about these differences and the distance between them that tells the cell where to splice. Right? That's one part of the cell figuring out where to splice a gene. But that's not everything. So here's the central dogma again. All of the exons are being taken out through splicing. Oops. I don't know how to draw. Right? All of the introns have been removed. You get a primary transcript or a mature transcript. It's turned into protein, translation. What else could happen here? This is the concept of alternate splicing. This is not always what's going to happen in the cell. What other ways could we splice this gene?
So the 2 that are spliced out end up being opposite.
So different order?
Yeah, change in order. Maybe not necessarily all of them slice.
Right. So you might wind up splicing out the first one, but not the second one. The second intron, that is. So you would wind up with not doing that splicing event. Because there's a lot of splicing that has to happen. Does every intron always get spliced out? No.
Then what's the point of splicing out introns in the first place if you're not going to get them all out?
Right. So here's the concept of alternative splicing. This is true. Not every gene is necessarily always spliced in the same way in the cell. Part of that's just chance. Because the mRNA is floating around in the cell, and if there aren't enough splicing factors or something's wrong, then some exons or some introns might not get removed. So here's a great example of how alternate splicing is actually useful. It's a little bit complicated, but it's okay because you're not being tested on this. Sex determination in Drosophila uses alternate splicing to dictate who's going to be male and who's going to be female. There's this gene, SXL, sex lethal, and it's alternatively spliced. If you're in a female, you'll see that exon 3 doesn't get included in the final transcript. So it splices from 2 to 4 if you're a female. How can it do that? Because there's a GU right there and there's an AG down there. So how does the cell know that there's also an AG right there? It just grabs a GU from here and an AG from there and sticks them together. So it splices out one intron and one exon. It could happen. Sometimes at low frequency, so sometimes alternate splicing they're rare. Like the cell forgot with that one transcript to do one splicing event. And not forgot, of course, the cell doesn't have a brain. But sometimes it's deliberate. So in a female, that third exon gets removed, and you make this protein sex lethal, wild type, normal protein, sex lethal. When the third intron gets left in, that third intron has a premature stop codon. So when translation's going, it hits that part and it stops making protein. Stop codon. No more protein. So it makes the first third or half of sex lethal and then translation's aborted. So you don't make the protein sex lethal. You make some N-terminal fragment of what might have been sex lethal. Okay, so at this point, flies that are destined to be males do not have sex lethal. Flies that are destined to be females have the full-length protein, sex lethal. Guess what? Sex lethal is a splicing factor. So this is going to have an effect on the splicing of something else. The protein, the gene, TRA, transformer. 2 exons. In females, when sex lethal is present, it binds over one of those AGs. So this binding site for the sex lethal protein, it covers a splice site so that splicing doesn't happen. And so you get the end of exon 1 spliced in the middle of exon 2, not the usual splice site. It's what they call a cryptic splice site, a splice site that's only used in certain situations. So here, females get exon 1 and the last half or so of exon 2, and that makes the full-length wild-type protein TRA, transformer. If you're a male, there's another premature stop codon in the first part of exon 2, so when 1 and 2 are spliced together like you would expect, there's that premature stop codon, half of the TRA protein gets made, it's not functional. So again, in males, you don't have the TRA protein, in females, you have wild-type TRA. Guess what TRA does? It's a splicing factor. This is the end of it, by the way, so I'm not going to say that again. It works on the gene double sex. So here's the double sex pre-messenger RNA. It's going to be spliced. In this case, the TRA protein binds to exon 4 and ensures that exon 4 is retained in the protein. And that makes a female-specific version of the double sex protein that has exon 4 in it. In males, exon 4 normally gets spliced out. So you don't get exon 4, you just get exons 3 and 5. It makes a different version of double sex. And then these proteins, the male-specific double sex and the female-specific double sex, go into the nucleus and turn on male-specific genes or female-specific genes. So at this point, these proteins are transcription factors, no longer splice factors.
Do you know what the ratio is of males and females that are?
In Drosophila,
Yeah.
it's half and half.
Oh, is it?
Yeah, and so what I didn't tell you is how sex lethal in the first place gets turned on. Yeah. There's sex chromosomes. So that's alternative splicing, just one story. That's how alternative splicing can come about. You get proteins maybe that direct certain splicing events to happen or not to happen, so that you leave out exons, leave in introns, and so forth. So the very valid question is, why alternate splice at all? Why have an inefficient process that's not always accurate? Maybe it has something to do with generating diversity. We have limited genome sizes. So this is a plot of how many genes are in different species and the size of their genome. So all of these lower organisms can have a lot of genes, but their genome sizes don't change much. And that might have something to do with the fact that it's not just how many genes you have, because the number of genes you have maybe limits something about your complexity. So how do you get more complex if there are only a finite number of genes? You can have different versions of each gene, alternate splice forms of every transcript to do different things. So comparing us to C. elegans, we have about the same number of genes in our genomes, but we're way cooler because we've got way more splice forms of those genes. So presumably that's why we've taken over the world, and we're not afraid of C. elegans, they're afraid of us. Or there's another way, this is just one other way to look at it, which is how much alternate splicing takes place in different organisms. What are the percentage of messenger RNAs that have alternatively spliced forms? In humans, it's something like 45% of our genes sustain alternative splicing. There's not just one way that splicing always happens, compared to many other species. So presumably alternative splicing is useful for diversity, for becoming better adapted to your environment, being more complex. Next time somebody tells me they feel like they're complex, I'm just going to tell them that they've got alternative splicing going on. Okay, so what does this have to do with next week's paper? So that's the basics of splicing. Switching topics totally, here are zebrafish. Wild-type zebrafish, recessive mutant, well, not a recessive mutant, a homozygous mutant, and a heterozygote for a mutation in this gene, Hagoromo, which I have been told by Wikipedia means dress of a goddess. Okay, and this is a Google Images picture that comes up when I Google Hagoromo. So this is zebrafish gene that's involved in pigmentation, and you can see what the effect is subtle, but you can see what the effect is when you get rid of the protein. So if you compare the striping pattern in this fish to that fish, you can see that there's a lot of jumbliness of the stripes. They're not lining up, they're not straight like they are in this fish. And even when there's just one wild type copy and one mutant copy in a diploid fish, their pigmentation pattern's still sort of screwed up. So this is a gene that's known to be involved in pigmentation in fish, pigment patterns, like cichlids, right? Lots of striped fish, lots of barred fish, vertical stripes, bars, horizontal. These stripes are stripes. If they go this way, they're bars in fish. So it's a gene that's known to have some role in pigmentation and patterning. And what we're going to see next week is that this specific gene in cichlids has lots and lots and lots and lots and lots of different splice patterns. So it's another correlation or causation paper, unfortunately, next week. They're trying to figure out, does the fact that Hagoromo in cichlids is extensively alternatively spliced related to the variation in color patterns, pigment patterns that in cichlids? Why is this at all interesting or important to study? Because changes in pigmentation can cause species formation. Here's an example from sticklebacks of males that turn red when they're ready to mate. These are the same species from populations that live pretty close to each other in Canada. Turn red when you're ready to mate, or turn jet black when you're ready to mate. Both males, both three-spined sticklebacks, different populations, do totally different pigmentation things when they're ready to mate. They have different signals that they send to females. And these populations are essentially different species now. In the last 100 years, this has happened. Blink of an eye in evolution. Because of these different colors. So females from this population now prefer to mate with black males. If you give them a choice between mating with this male or that male, females from this population will only mate with these males. And females, this female, for example, would only mate with that male, not with black males, despite the fact that they're all the same species. So there's been a change in male pigmentation and also a change in the female eye, like the Opson paper we read today. So there's something that's changing in the vision of these females and the pigmentation of the males, and that's driving speciation. So that's how this to the cichlids. All of those species of cichlids, that poster shot that I keep showing with all the different species of cichlids, blues, yellows, greens, reds, stripes this way, stripes that way, that's why these researchers are interested in pigmentation. Because it might be that they could understand how the evolution of pigmentation has caused the formation of all of those species. So this is basically what they're going to do. They're going to try to figure out how many different ways is the Hagoromo gene spliced. Is there just one alternate form, or are there 2, 10, 50, 100, 1,000 different ways it can be spliced? And then they're going to try to correlate that number with speciation rate. So if the hypothesis, I suppose, is if Hagoromo is involved in speciation, then the more splice forms you have, the faster speciation occurs. We'll see. Okay. So all they're doing in this paper essentially is PCR, polymerase chain reaction amplification of the Hagoromo gene. They just want to see how many different ways does that mRNA get spliced. So with PCR, you start with template, and you add some primers, and you make lots and lots and lots of copies of that original template so that we can see them on a gel. What these people are doing, because they're not interested in genomic DNA, they're interested in messenger RNA. You can't do PCR with messenger RNA because messenger RNA is single-stranded. To do PCR, you need 2 strands. It needs to be double-stranded nucleic acid. So messenger RNA is just a single strand. So what do? What do? They make cDNA from messenger RNA. So they take fish, they pull out all of the messenger RNA. How do you do that? Okay, so I hope I know how to write this. Reverse transcriptase, close enough. I think my I is inside the tail of my P, but whatever. So how do you get all the messenger RNAs out of a cell? Just the messenger RNAs. And nothing else. No other DNA RNA, tRNA, ribosomal RNA. It's that poly A tail. So if you have something that's got a lot of T's in it, you could do some sort of like a Western blot. Or not a, well, the pre-Western blot, the IP, the immunoprecipitation. You could have a column of beads that have poly T, just T's, and then all the messenger RNA with all their poly A tails stick to the column, all the other nucleic acids go through, and then you wash off just messenger RNA. So you can extract just the messenger RNA. And that's what takes us to the first step here. So you have messenger RNA with a poly A tail. You add a single-stranded primer to that, and then you add that enzyme, was mentioned, reverse transcriptase, which copies C, copies RNA, makes a DNA strand. So you get an RNA/DNA hybrid. Half DNA and then the complement, half RNA, the complement to that is DNA, so it's double-stranded. Then you can use that as a template in PCR. You make lots of copies, and from that really tiny amount of RNA you started with, you make lots and lots of copies of double-stranded DNA products. So that's what you wind up with at the end. I should mention that this RT-PCR, reverse transcriptase PCR, is different from a different type of RT-PCR. And I always get confused about this, so I'm going to tell you now so you get confused. There's another type of RT-PCR that's real-time PCR. So there's reverse transcriptase PCR and there's real-time PCR, and sometimes people call them both RT-PCR. So anytime you see RT-PCR, just be careful that you're noting which of the 2 different RT-PCRs you're talking about.
I thought real-time was the Q.
Sometimes real-time PCR is also called qPCR, quantitative PCR.
So
That's a better thing to call it than RT.
Sometimes when it's RT, it's actually supposed to be real-time.
Yeah. Just because people are ignorant, and sometimes I'm ignorant too. I'm not trying to put this on anybody else. So sometimes I might write RT-PCR instead of qPCR when I meant qPCR, but that's less ambiguous to do qPCR. Anyway, so this is reverse transcription. Okay. One other technique that they're going to use is in situ hybridization. This is not fluorescence in situ hybridization, which we've talked about before. This is just straight up in situ hybridization, or RNA in situ hybridization. And I tried to find a good picture of this. I can draw it, which I'm going to do, but I couldn't find any good pictures of this on Google Images. I don't know why. So if there's a gene that's involved in pigmentation, where will you find it expressed in the body of a fish? Is skin, scales, or the hypodermis, something just underneath the scales. So the reason that people do RNA in situ hybridization is, well, does anybody know? What does in situ hybridization do? I'll tell you if you don't know, but has anybody done this before? So RNA in situ hybridization tells you where in a tissue a specific messenger RNA is being expressed, transcribed. So if you take a microscope slide with a section of a fish, let's see. Oh, let's just say you took a longitudinal section of a fish. That's not a wrench, by the way, that's a fish tail. Look, here's the spot where its eye would be. Okay. So we're going to say if this gene, Hagoromo, is involved in pigmentation, that it should be expressed in the skin. So actually what I should do is section the fish that way so that I'm looking through its body. And we expect to see Hag in the scales, in the skin, just underneath the skin, on the surface of the fish. So what we could do is take that section of a fish, put it on a glass slide. We want to find out where are the messenger RNAs for the Hagoromo gene. So you take a single-stranded RNA probe. This is very similar to fluorescence in situ hybridization. It's fluorescently labeled, or somehow labeled. And it's complementary to the Hagoromo messenger RNA. So if the Hagoromo messenger RNA has A, T, A, G, C, except, what did I screw up? I always do that. This is messenger RNA. U, AUAGC. Then we'd expect somewhere in this RNA probe to have UAUCG. So if those messenger RNAs are in the tissues, in the cells that are just underneath the skin, and nowhere else in this tissue section, then what we do is we take that microscope slide, you permeabilize the tissues, you fix the tissues so that you can get molecules into the cells, and all the RNAs and DNAs, everything stays put on the microscope slide. Then you add this probe, and everywhere there's a match of the probe sequence to the messenger RNA, you get hybridization, double-stranded RNA-DNA complexes forming. And that's where you can actually see by fluorescence, because all these are fluorescently labeled, you can see which cells have transcript for whatever is complementary to the probe. So in a nutshell, this is just used to figure out which cells in an organism contain a messenger RNA that you're interested in. Now the problem with this is that the probe that you use might bind to more than just the one gene. Especially if there are lots of genes that are closely related, like a gene family. They're mostly the same sequence, they're just slightly different. So there's a control experiment you have to do, and this happens in this paper, so I want to point this out because it's important to know for in situ hybridization protocols. This part down here. Antisense and sense probes. What does that mean? This is an antisense probe. Except I really can't spell now. Holy crap. Antisense probe. That's the one that actually hybridizes to the messenger RNA you're looking for. What would a sense probe be? What color should I pick? So a sense probe is the complement to the antisense probe, which is basically the sequence of the original. Why would you do that? So in one experiment, one glass slide, you put your sense probe, and you find out where it binds. And the other slide, the next tissue section over in the body of the animal, you apply the other probe. Antisense probe should hybridize to the messenger RNA and tell you where it is. What should the sense probe do? Does it have anything to do with the Hagoromo messenger RNA sequence? No, it's the same sequence as part of the Hagoromo messenger RNA, so it shouldn't bind anywhere. So this is the negative control. Because there was DNA that's complementary to the RNA probe. Because DNA is double-stranded, so there is guaranteed in every cell in the nucleus something that will hybridize both to a probe, the sense and the antisense probes. So this is the sense probe is the negative control to make sure that you know what the background level of staining is. There's always going to be a certain amount of staining of every cell. What we're looking for is where are the brighter cells that show us specifically where the messenger RNA is, and that's going to be specific to the antisense probe.
So you mean the background staining in the same cell or among
In all the cells, there's going to be some, and usually you don't see it in manuscripts. Usually they subtract out the background so you don't see it. But you do want to know that your probe, wherever it's hybridizing and showing you there's something there, that it's mRNA that it's binding to, that you're seeing the signal from, and not another nucleic acid that's similar, like DNA. So that's what they mean for the paper next week, probes. And last but certainly not least, this is important and kind of confusingly written in this paper, I think. So they talk about speciation index in, I think it's the last figure. And what that means is the number that they assign, it's called the speciation index, is how long it takes a particular group of fish to double in the number of species. So you've got oh man, what the hell am I doing? I don't know how to draw phylogenetic trees anymore. So here's two groups of fish. Here's one clade group, and here's another one. So we've got clade 1 and clade 2. 7 different species. 1, 2, 3, 4, 5, 6, 7 current species. So let's say we've got some sort of a timeline on here. So this is present always at the tips of the branches. Let's say, what's the acronym we're using? MYA. So we'll say this 1 MYA, 1 million years ago, 2 million years ago. What's the speciation index of clade 1? We had, we have 4 species currently, so where's the point in time when we had 2 species in clade 1? It's wherever there are two branches. So about 1 million years ago, there were 2 species in this clade. Present, there are 4. So in 1 million years, they doubled. So the speciation index here is 1 million years. And then in this other group, well, let me see. Let me fudge just for a second. Let me add another species to make the math easy. Okay, so they had two species there 300,000 years ago. There are 4 species currently, so their speciation index is something like 0.3 in the last 300,000 million years. So in the last 300,000 years, the species number has doubled. So those are what those numbers represent. And again, what they're going to try to do is correlate which of these species has lots of different types of Hagoromo protein, and is it the groups that speciate faster that have more versions of this protein? Presumably more visually diverse, and it might be that visual diversity, like in the sticklebacks, the red versus the black males. It might be the fact that if there are a lot of species and they're all visually different, that leads, that Hagoromo is producing different pigment patterns that lead to speciation. So that's the goal of the paper, is to figure out whether or not that correlation exists.